Next up, I'd like to welcome our science director, Eric Diehl, to the stage to talk a bit about his work with our collaborative research network and our next steps. Eric has worked in epigenetics research for over 10 years. He completed his undergraduate and PhD in biology at the University of Western Ontario. His thesis involved identifying epigenetic changes in a mouse model of fetal alcohol syndrome, spectrum disorders. He then completed a postdoctoral fellowship in the laboratory of Rosanna Wexberg at Sick Kids in Toronto, focused on development of epigenetic signatures to aid diagnosis of neurodevelopmental disorders like TBRS. He is also employed as a diagnostic laboratory geneticist at Sunnybrook Hospital in Toronto and lives in his hometown of London, Ontario. So I'd like to welcome Eric.
Thank you, everyone. I've been really impressed by all the science we've seen so far today. I think it's really a testament to everyone's hard work and dedication, and we as the community really appreciate that. So we'll be the first to say thank you again and again, as we will do over the next couple of days. So the function of this talk is a few things. So we have a panel discussion coming up next right after the break, so I kind of want to serve this as sort of an introduction and primer to sort of facilitate and prompt the already great discussions that we're having. But I also sort of want to introduce myself as being relatively new to the community and sort of give some updates on what I've been doing, what my role is, and sort of the progress that I've made so far. So what am I supposed to be doing in this role of science director? So the main thing is to sort of maintain knowledge, to engage with the patient community, look for current and new opportunities for patients to participate, sort of build network and execute engagement, and then sort of a big piece is to conduct a comprehensive gap analysis and identify any missing components in our research roadmap to sort of help us push forward towards interventions or therapies. So Kit gave a great introduction on me, so I'm going to kind of skip through that. Just to say that I'm really interested in epigenetics, have been for a very long time, have a lot of interest in mouse. I've really enjoyed seeing all the mouse talks today, and diagnostics, VUS classification are sort of where my interests and passions sort of lie. So a gap analysis is a 4-step process in order to sort of see, basically where are we and where do we want to go and how do we get there. So the big thing that I've been doing is these first 2 steps, which are analyzing the current state and identifying the ideal state is sort of an easy one. So I think we, as the community, as patients and families, have set the ideal state as, a FDA-approved therapy or intervention for TBRS that improves primarily cognitive outcomes, but there are, other components of that as well. And Carrie's going to be speaking more on the updated patient priorities a little more tomorrow. So in order to do that, I've been meeting with many of you and with many of the other researchers who are joining us online to sort of get a sense just of where we are as a community, where we are as a field, what we know sort of about TBRS and DNMT3A. And so one of the main functions of this talk I want to do is sort of give all of you the benefit of all of those conversations, sort of tell you all sort of where you agree, where we disagree, and sort of where we see as the path forward. So I'm going to be giving another version of this talk tomorrow to the families. I'm going to be targeting a little more stuff to each of these audiences a little differently. So we talked a lot with all of you about the different aspects of TBRS that are being researched. We definitely have a consensus that therapeutics are the goal, that more basic science is needed, and that the community is really poised well to support this. And everybody sort of universally agrees that the biorepository and the registry are sort of the best things that we can be doing as a community to support research. And I think we see that evidence certainly in Kate's work. So where do we agree? We agree on an enormous amount of things, I will say. So everybody agrees that the collaboration within the community is outstanding. Generally, everyone's very happy with the direction we're going. The research progress has been quite fast, and I have to say, just as an aside, coming from an outside perspective, coming into the community, I have to say I'm just incredibly impressed with the pace of research that's happened since TBRS was first described. And just, being at the CZI meeting a couple weeks ago and seeing where other patient organizations are, it's really a testament to the work of everybody who's involved that we sort of are where we are. So I just want to talk everybody up and say that it's really an amazing amount of work that's being done so far. something that we came up with, that sort of came up is, therapeutics being a goal, and like what Kara will talk about as well, is that, therapeutics specifically targeting intellectual disability and other cognitive phenotypes are a main goal, but I think something we sort of all agreed on in our conversations that, defining some short-term wins is a good strategy in order for us to work towards things that are a little more achievable in a much nearer time frame. So things like Kate was talking about addressing management guidelines is something we can do in the nearer term, and that will have a lot of really positive benefits, and that's something families are hoping for as well. So talking about some areas where we see some, I don't want to say disagreement, because I don't think that's really the right word, but just where we see differences sort of in the way forward for TBRS and DNMT3A research. The biggest thing I noticed, when we were talking, is that, sort of the vision for what a successful therapy looks like are really different from different researchers. So a lot of people see drug repurposing as the best option. A lot of people see drug screens as where we should be focusing. Gene therapy, rational drug design, there's sort of many different opinions on the right way forward and ultimately what's going to be successful. There's also was a lot of discussion. I asked everyone sort of about whether we should be looking to target DNMT3A pharmacologically or whether we should be looking at downstream other molecular pathways that maybe are affected. And this sort of really reflected, I think, people's views on whether and to what extent DNMT3A acts early versus has some lasting effects that go later into life. And I mean, all of your talks have sort of really talked about this today as well. So final thing I think where there was some differences was whether there's benefit to grouping TBRS in with the other OGID conditions and sort of going into maybe looking for drugs that can work on all of these conditions. Some people think, yes, why would we not do that? Other people think, we've done so much work to define TBRS as its own entity, and molecularly these are quite distinct conditions that we need to focus on doing that. And on that one, I'll just say that we're sort of aware of that as a community, and there's a lot of work we're doing to work with the Overgrowth Syndromes Alliance to sort of, I think we're being smart in terms of how we're doing that, and I think that one sort of is not necessarily a dichotomy, that we're sort of able to get the benefits of both of those things. And the important thing from my perspective looking at this is that none of these things require decision making now. We're not at a point where we as a community need to say, okay, we have X amount of dollars that we need to put down and work on one therapy. We're sort of very happy and very encouraged to be supporting all of these ways forward because we have a lot of work still to do in terms of basic science to see which sort of path we want to go down and which one's ultimately going to be successful. So to sort of fill in the progress that I've made on this, so I've talked to many of you, I'm meeting some of you for the first time today, so I'm hopeful I can have some more meetings with those of you I haven't got to yet, but sort of identifying the ideal state. There's two major components to that I think we see. One is to have TBRS as a well-characterized condition with diagnostic risk and management guidelines that we've been talking about today. And the other, ideal state is to have a therapy or intervention, the nature of which is still sort of unknown. So the gaps to get there, the gap towards getting TBRS better defined is just to, further define it. And then, so the knowledge gaps are sort of what to target and when in terms of a therapy to sort of distill down a very complex topic to a couple of words. So on that first bit, I want to credit Kate with sort of defining these categories for me when we were having our meeting, and I know this is sort of reflected in her talk. But we still have a couple of issues in defining TBRS. One is the VUS issue that we've talked a bit about. So as I was looking and helping Kate with that work she presented today, we have a challenge in that a lot of variants that can, and in my opinion should be being classified as likely pathogenic or pathogenic, are being classified as VUS. And there's a lot of divergence in how clinical labs are classifying them. And that's due, as we've been talking about, to the clonal hematopoiesis issue, and also the ClinGen score. Its haploinsufficiency score is a 1 right now. A 3 is what is needed for a clinical lab to define any deletion as pathogenic just by default. So we don't have that yet. The other thing is, of course, that we have more missense versus, the reason for that is because, as we've been talking about, we have a preponderance of missense variation in this gene compared to frame shift or deletion variation, and so because of that, ClinGen has our score at a 1 currently. And that's just to reflect the uncertainty that we've been talking about. So we have some plans underway to address that. I'll go over that tomorrow because I'm addressing that specifically to the families. And then DNMT3A function itself, you know, we've been talking an enormous amount about that. I'm not going to try to summarize all of the work that we've been talking about. And then, of course, the clinical features and the phenotype, again, what the work that Kate and others are doing to sort of fully characterize the life history of TBRS. So the goal then is to get to a management strategy, you know, to see sort of what challenges patients can expect, what they should be screened for, what therapies are available, and get to surveillance, guidelines, incidence, natural history, and phenotype. And I think we're sort of set out on a path in order to do that that looks really good. So just to talk about that missense question a little bit, I added this slide during Kate's question period as I was talking to Harrison, so thank you, Harrison, for making me think of this. But there's this image I really like from a textbook I have that shows, you know, in a lot of conditions we think of, you know, trisomy 21 is viable, Turner syndrome is viable, but what's actually the case is if this iceberg represents individuals who are born being the tip of the iceberg that's shown above, there's all of these other chromosomal aneuploidies that are lost prenatally. So we don't get to see those. They're lost, and we think, you know, oh, you know, Down syndrome is viable, but there's actually an enormous amount of those pregnancies that are lost prenatally, and the same is true for Turner syndrome. So I'm just wondering, and we can talk about this later, just sort of posing a hypothesis that perhaps deletions or frameshift variants fall into this sort of under the iceberg thing and they're being lost prenatally, and maybe we could get at that question by looking at, you know, some of these product of consumption biobanks maybe could help us test this hypothesis. So something to think about. The other big gap is then in terms of therapeutics, what to target and when. So this is all of the work everyone's engaged in, you know, identifying affected cell types, identifying potentially druggable targets. I can't even cover all of the bullet points that encompass all of the work everyone's doing. So there's an enormous amount of work going on sort of to see what potential targets there are. And then so sort of from a clinical trial perspective, getting enough data as we were just talking about, and there was a really great primer for this showing the amount of work necessary to get towards a clinical trial. There really needs to be a lot of preclinical data that in order to support that, and that's sort of where we are right now and what we're engaged in right now. And then again, importantly, and this is sort of the point I'll make mostly to the families tomorrow, is that, you know, this isn't an opportunity cost decision. It's not we're missing out on one thing by focusing on another. This is all work that we're as a community really happy and excited to support, and we see all of these options as very viable, and we want to support everything as much as we can. So what are the goals for the community then? In the short term, you know, we want to facilitate and encourage use of the biorepository, and Citizen is something we're encouraging the families to sign up for to do that. We want to get targeted funding to everyone as much as we can, and I'm going to go over a bit more of that in the panel discussion to sort of talk about how we want to do that. Address the VUS problem, sort of as I was talking about. I'm wondering maybe if there's a guideline paper we can do that might help to sort of reduce the amount of unnecessary VUSs that we're generating. And then in the long term, something else I sort of want to bring up talking about is whether the development of specific working groups may be helpful for those of you that are interested in specific aspects of therapy. Like maybe we can have a you know, drug repurposing working group and sort of be focused in, you know, individual, you know, a few people, and bring in people who are experts in other conditions to sort of do that. And so we're wondering if maybe that'll be a strategy that'll work for us as well. So yeah, after all of that, I just want to say sort of thank you again for everyone's work. It's been a really a pleasure to become a part of this community. And yeah, I'm happy to take any specific questions right now, but I really sort of view this talk as a primer for our panel discussion that we're going to be having after the break. So I'm hopeful that we can, you know, have a great conversation or continue to have a great conversation as we've been doing at this meeting. So thank you.